Okay, good morning. Welcome back, everyone. Welcome to the last pre-conference, pre-workshop day of the conference. Very happy to start off today's activities with a keynote lecture by Professor Carolyn Uhler. Dr. Uhler is a professor at MIT in the departments of EECS, Electrical Engineering and Computer Science, and the IDSS, the Institute for Data Systems and Society, and also in the past few years became the co-director of the Eric and Wendy Schmidt Center at the Broad Institute, a joint venture between Harvard and MIT. Caroline has many awards and accolades that I'll highlight just a couple here, including a Simons Investigator Award, a Sloan Research Fellowship, an NSF Career Award, and many others. But you probably know her work best for doing really innovative fundamental research at the intersection of statistics and machine learning on topics as diverse as algebraic statistics, graphical models, causality, autoencoders and neural networks, and applications in high-dimensional comp bio, genomics especially. So we're very happy to have Dr. Uhler here today to talk about causal representation learning and optimal intervention design.
So I'm very, very excited to be here. So thank you very much, Daniel, for this very kind introduction, and to all the organizers for inviting me to give this keynote here today. So a lot of my work, as Daniel said, has been at this intersection between statistics, machine learning, and then really looking at applications from the biomedical sciences. And in particular, what has really fascinated me is, how the biomedical sciences and particular questions there can really motivate new fundamental developments in machine learning and statistics. And what I want to do in this talk is tell you a little bit about this fascination, hoping to invite many more of you into this field of actually looking at biological questions and then developing the foundations of machine learning that are really needed in order to make progress on these questions, which is really at the center of the Eric and Wendy Schmidt Center that I co-direct that Daniel mentioned, where we're really trying to develop this two-way street between the biomedical sciences and machine learning. So not only, what computational biology I think has done very well is bringing in machine learning methods into biology to solve questions there, but in particular going the other direction of having the biomedical sciences drive what are some of the foundations of machine learning that need to be developed in order to then really have an impact on biology. And so I think that two-way street will maybe become a little bit more clear as I go through this talk. So the biomedical sciences are really seeing a data revolution where, it is possible now to, in a single experiment, profile millions of cells. And you can do this using sequencing technologies or imaging, like what you see here. You can actually do imaging at the single cell level. You can do imaging at the tissue level, at the organ level, even at the whole human body level. And you can get all these very different types of data modalities from, different types of experiments. And so just to make it a little bit more clear about the types of scales that we're looking at in biology. So this is just the amount of data that was produced in one single research lab. Okay, so that's, and again, this is only genomics, so only sequencing-based data, no imaging. So just the Broad genomics platform alone last year produced 80 petabytes of data. So now if I can trust what I can find online about Twitter and Netflix, and actually this curve looks very similar for Twitter. Okay, so about the same amount of data that they generated last year, and also about the same growth that you see here. And compare these 80 petabytes of data to, say, for example, the whole Netflix movie corpus, which is only about 60 petabytes of data. So, despite, and again, if we take also all the imaging data sets are generally much larger than sequencing data sets. So, all of that is not included in anything here. Okay, now despite, seeing this huge, the biological sciences basically overtaking any other field in terms of data generation, it is still application areas like, recommender systems or online advertising or self-driving cars that are some of the main motivators for the developments in, the foundations of machine learning today. And so what I'm suggesting here is that, the biological sciences should really be looked at very carefully, and that they might actually lead to different types of questions that are important that you wouldn't really necessarily get to when you care about, say, recommender systems. So for example, in a recommender system, often just prediction accuracy is often sufficient, right? If you know that someone is interested in ice cream, you just have to figure out that this person might also be interested in sunscreen, but you don't really care about why that's the case. Whereas in the biomedical sciences, you really do care about the underlying mechanisms, right? We do care if you want to develop drugs. You want to understand how are different proteins interacting, and what is, which protein do you have to, say, down regulate in order to move a cell from the disease state back to the normal state, etc. So these are totally different types of questions for many different applications in the biomedical sciences, where you do have to go a step further and really get at the underlying mechanisms or the underlying causal relationships. Now, how can you get at causality? And we've seen so many amazing talks here already. And in particular, in the biomedical sciences, we have this huge hammer, which is interventions. And in biology, they're called perturbations. Where, how do interventions help you, right? If you want to understand here that warm weather or sunshine increases ice cream consumption, well, you, for example, take, or you want to understand whether it's this way or the other way around, you take a gloomy day, you go out and you give out a lot of free ice cream, right? And you see that doesn't change the weather, therefore ice cream consumption is not upstream or not causal of nice weather, right? So this is the power of interventions for you to understand what is actually downstream and what is in fact upstream. And that is the hammer that we have available in biology, where you can perform very, very large scale interventions. So you can genetically, through CRISPR, you can genetically intervene on any gene you like and see what the effect is of that. So that means that you can figure out which gene is upstream of which other gene, right? Through similar logic, or you should be able to figure out through similar logic as I just went through here, which gene is actually upstream of which other gene. You also have other types of perturbations that are chemical perturbations, right? I can use drugs to perturb single cells and see what happens. And so we have in the biomedical sciences, I think we have this really nice kind of problems that are always coming from having very, very large scale multimodal data sets. So sequencing, imaging at different scales, and also another modality, I'll call it here modality, which is large scale interventions. And so I think this really calls then for a framework where, maybe we need to be able to somehow develop a framework where we can integrate all these different modalities and also, in fact, use interventions in order to get at the underlying causal mechanisms. Now, you could think that maybe current representation learning methods might already be sufficient to think about, causal representations. And so here I just did a very simple experiment, taking this a step further. I asked DALL-E to generate an image of, an ice cream stand where it gives out free ice cream. And you can do this many, many times, and you'll always get good weather in the background. Okay, so even though I tell it that I'm giving out free ice cream, it will still always do good weather in the background, although, the distribution of weather should not change, right? When I'm giving out free ice cream. So it shouldn't suddenly, so what it is doing is kind of conditioning on actually seeing ice cream in here and not doing an intervention on actually giving out free ice cream. So current methods, current representation learning methods are just not geared towards actually getting at underlying causal mechanisms, and really do very, very poorly when you give it, some intervention, or you tell it to produce data from a particular intervention. Okay, so that really motivates that we do need more fundamental theory here to try to integrate multimodal data and then also interventional data. Okay, so how do we think about mechanisms in biology? So traditionally, what one has thought about as mechanisms is these large-scale networks where you have all these different genes or proteins, and then you have this large network of these are causal effects or, regulatory relationships of this gene up or down regulates that gene. And just to give a little bit of background, how you should think of this is a small network, but you should think of in humans, we have 20,000 genes. Okay, so think of a large directed network on 20,000 nodes. That would be a gene regulatory network. And of course, we'll develop the foundations for actually thinking about these networks using, in a causal way and also in a probabilistic way. And I don't have to, for this audience here introduced causal structural equation models, but this is the framework that I'm going to be using. So every node is a random variable, and it is a function of its direct parents and some noise. And this noise here, these epsilons, is how the stochasticity is being introduced. Now, the nice thing about these causal structural equation models is, of course, they also define the interventional distributions. And we've seen wonderful talks here about interventions, soft and hard interventions. This one here, I'm just writing down the do calculus that is often used, which would represent hard interventions, but I will be talking a lot also about soft interventions. So here in this particular hard intervention, you would just set a particular node to a particular value. So for example, for genes, I would knock out that gene and set it to 0. And then I would see what happens to the expression of all other genes. Okay, so if you do an intervention on, say, X2, set this to 0, then only X4 changes, but of course, what is upstream doesn't change. So, you know, interventions or interventional distributions or interventions really define probabilistic operations that are not just conditioning or marginalization, which, you know, standard representation learning methods are pretty good at doing, just marginalization or conditioning. But you see here very clearly that this is not the same, can sometimes lead to marginalization, can sometimes lead to conditioning, but it's certainly not the same operation. Okay, so I'll talk a lot about interventions in this talk and also about multimodality, and maybe I also just a little bit more about interventions. So I mentioned genetic interventions where, you know, you go in and you, for example, knock out the gene, so set it to 0. So that would be a hard intervention, but literally all these interventions are generally soft. And I think I also during the talk just want to motivate always like questions that come up that I think are important that we haven't worked on enough yet. You know, generally, yes, the knockout is a hard intervention, but often, you know, what you see is that the knockout happens with a particular probability. So in some cells, it will have happened, in some cells, it hasn't happened. So that in some sense is again a soft intervention. But we don't have a lot of models of useful soft interventions. So either we have theory that is just holds for any soft intervention, or we often look at soft interventions, which are shift interventions. So I'm just changing the mean. And, you know, I'm as at fault here as everyone else. So my papers also often just looked at, you know, shifting the mean as a soft intervention. But I think there are so many important classes of soft interventions that we should be looking at that where we just don't really have the theory yet for thinking about them. Okay, so let me move on from here and talk about the motivation of what are the types of problems that, you know, we really ultimately want to solve with these perturbations. So this will be like, you know, has motivated or is motivating much of my lab's work. And generally, it's just a really important, like a lot of problems in the biomedical sciences fall into this kind of question here, which is engineering cell states. Okay, so the human body, we have hundreds of different cell types, right? We have neurons, we have skin cells, we have lung cells, we have fiber, we have connective tissue cells, et cetera, heart cells, et cetera, et cetera. Now, what would be really amazing is if you could take any cell and make another cell out of it, right? In particular, could I take a skin cell, which is very easily available, make a neuron out of it, and then test all the different drugs in a person-specific way, right? And I can throw all these different drugs at these neurons. You cannot take just neurons out of the brain, right, to do these experiments. So that would be really helpful for personalized medicine. Of course, you can think of applications, regenerative medicine. You can do rejuvenation, take out cells, put them back into the body, et cetera, in different states. Now, the exciting thing in biology is that we know that this is possible. Okay, so here, for example, fibroblasts are like the most abundant cells. They're the connective tissue. So they're the most abundant cells in our body. So, you know, Yamanaka, who got the Nobel Prize for this, has shown that you can take these fibroblasts and actually reprogram them. So genetically, or in this case, chemically perturb them and make induce pluripotent stem cells out of it, out of which you can then make a new mouse. Okay, for example. So this is like taking a differentiated stem cell type, going back, de-differentiating it, and then actually making a mouse out of it. So out of these cells, you can actually make any cell type you like. Okay, so this is possible, and how they did it is by really careful experiments, doing a lot of different experiments, and figuring out which are the right combinations in order to actually move one cell type to another cell type. Okay, so now how large is the space and what are we thinking about here? So often, so we have, as I said, 20,000 genes. 2,000 of these genes are special. They're called transcription factors. They have important roles in regulation. So often people experiment with these 2,000 genes. Okay, so these 2,000 genes, and then you do any combination of them. And so here they found the right combination of 4 transcription factors out of these 2,000 that actually move you from this one cell state to the other cell state. Okay, so the space is huge, right? 2,000, choose any combination. So choose 1, choose 2, choose 3, choose 4, etc. So that's the space of possible perturbations that you can very, very easily perform on any cell type. So we have hundreds of different cell types. Okay, so the space of possible experiments is just extremely large. That's one of these challenges. And you would really like to be able to go from any cell type to any other cell type using any of these combinations of interventions. And that's just, so that's just, interventions using these transcription factors. So that's kind of what we have been modeling with these shift interventions. So I'm making this gene be more expressed. Okay, so you choose one of these 2,000 genes and you're adding whatever that product is of that gene. And then those are the interventions that have been used for these reprogramming events. Now, let me just give you a completely different application just to show again how important these problems are. Cancer immunotherapy. So what you want to do there is, our own bodies are capable of killing cancer cells. The problem is that, so it's the T cells, it's the soldiers in our body, they're able of killing cancer cells. But the problem is that the cancer cells send out signals to make these soldiers tired. Okay, so they actually move them to this terminal exhausted state where they cannot kill cancer cells anymore. So now what cancer immunotherapy tries to do is take your own soldiers, your own T cells, and change them so that they don't go into this state, but actually, remain these soldiers here of the immune system so that they can continue killing cancer cells. Okay, so that's the whole promise of cancer immunotherapy. And so we did this challenge here where, again, what we need to do is we take T cells. So we did this in the mouse, right? So we take these soldiers of the immune system, so these T cells, and we try to ask, well, can you come up with, and we give you some perturbations. We cannot do, in this case, we looked at genetic perturbations of all 20,000 genes. I can go and knock out gene 1 or gene 2 or gene 3, and then that moves the cell to a different state, right? And so now, I cannot go and in a mouse actually perform all 20,000 experiments and then see how active is this, how well can this cell actually be killing cancer cells. So we asked, given, say some 20 interventions, can you then, the whole community, can you predict which intervention would be the best one in order to move these T cells from this terminal exhausted state away to, either progenitor, effector, or cycling states. So all of these states are good states to be in. Okay, so these are the kinds of experimental design questions that we need answers to. I give you some interventions. Can you then predict the effect of other interventions that we haven't seen? And so how you should think of this always is like, what we get as a measurement is a measurement over all our 20,000 genes. Okay, so per cell, I give you the measurement of all these 20,000 genes. And I give you this measurement of 20,000 genes for after you do a perturbation. So say you knock out gene 1 and I give you 1,000 cells of what happens when you knock out gene 1. And this is just a picture in 2 dimensions instead of showing you the distributions in 20,000 dimensions, which I cannot, right? I'm just doing a nice visualization in 2 dimensions. So every dot here is a cell. And this here, I'm just showing you the observational data, but also for a knockout, you would see that maybe some cells are more in this state or in this state or in this state, right? For a different knockout, maybe that moves the cells mainly into this state. And so what you want is to find knockouts that really move you over, right? That where you have as few cells as possible ending up in this state, but they all end up in this state here. Okay, so that's how you should think of these types of questions. So the challenge is really that the space of possible perturbations and also relevant context. I mean, here I gave you an example of T cells. Here I gave you an example of these connective tissue cells. You may care about any cell type, right? And you want to move those cells or any diseased state, and you want to move them back to the normal state. So those are the kinds of big underlying questions that we care about. Okay, so how do we go from here to actually doing some, or what types of questions does that lead to in causality? Okay, so if I were able to give you this whole underlying network or some causal representation, so some causal structural equation model, say, that represents, what is going on in a cell, well, then, you could actually predict what happens if I do a particular intervention, right? And so then maybe you could figure out what is the best intervention in order to move a cell from one state to the other. Okay, so you could think that, of course, in biology, I don't know this underlying network. That's the whole difficulty. So maybe, because I have so much such 20,000 dimensional cells, right? In every experiment, I can actually give you 1 million of these cells. So maybe if you have 1 million cells, maybe then you can learn this underlying causal network. And then from there, try to predict what is the best intervention to do in order to move these cells to a different state. Okay, so that leads to causal structure discovery, and I have to have it in here, given that we're here at CMU, with all of the wonderful foundational work that has been done and has really, led to the birth of this kind of field of trying to understand how much can you learn about the underlying causal structural model just from observational data, so just from data on the nodes. So that's this, amazing book by Espertus, Gleimer, and Shines, and in particular, the first algorithm that was able to do this from observational data under certain assumptions. And so that's a PC algorithm. And any algorithm for causal structure discovery, it's just a hard problem, right? We'll always have to make some assumptions, and they're known as certain faithfulness assumptions. And so what these assumptions tell you is somehow that it's very hard to learn this causal graph if you have cancellations in it. So let me just go through this example. So for example, in the PC algorithm, it's all based on conditional independence testing. So, if this is the graph, so gene 1 down regulates gene 3, and gene 3 up regulates gene 2, and gene 1 up regulates gene 2. Well, if you choose these edge weights in certain ways, it could be that if you upregulate gene 1, then it down regulates gene 2 through this path, but it upregulates gene 2 through this path. Right? And so these two paths could cancel each other out. And so it could look in the data as if, expression of gene 1 and expression of gene 2 are independent. Right? And so if it's independent, well, then you would remove this edge and you would even infer wrong edges. So that's a faithfulness assumption that tells you that, paths shouldn't cancel each other out. Now, the problem is that paths, when you have data, of course, it's never exact, right? So the paths also cannot even approximately cancel each other out. And then it becomes kind of problematic, right? Because you might have many different possible cancellations, right? And so what we have been analyzing now already multiple years back is that, this is really problematic because you can actually, and you will, in fact, have these cancellations happen very often. And it's not the exact cancellations, but it's these approximate cancellations, which will just happen very often. So, and here again, I just want to point out also a lot of open problems. So I think the problem is here that you do a lot of tests, right? And every test somehow can give you a wrong result, and this will, have a big effect downstream. So questions are just like, how can you do, like, what are algorithms that can get away with the least number of independence tests? And those should generally do better because you can have less violations. And so, I will not go into this because that won't be the main point of this talk, but I think that there have been a lot of developments in terms of trying to maybe do something with orderings. And I'm sure, in the tutorial that you had on Monday, there were probably also quite a lot of talks also about ordering-based approaches, where maybe you need, and we prove that you definitely need weaker assumptions than, what you would need in this faithfulness, just because you are doing somehow less conditional independence tests for every one of the orders. And you can extend this to interventional data. And here we're really building on Frederick Eberhardt's, notions of or introduction of what it means to be a soft intervention, a hard intervention, how we should think of it, etc. But, and I think this is the main thing that I want to say is just that, yes, we do have algorithms that computationally scale to graphs with thousands of nodes, where we can do structure discovery, on thousands of nodes. But the problem is just performance-wise, they're very poor. And and in some sense, independent of which algorithm you use, they just make so many mistakes in terms of learning the underlying causal structure model. You can apply them, I think, or we always feel like, even if we have 100,000 cells or 1 million cells, we maybe apply them to graphs with about 100 nodes, but we generally don't go higher than graphs with about 100 nodes. Okay, so yes, so they have been like really amazing in terms of learning smaller graphs, and you can apply them to large graphs, but in terms of actually getting the correct graph, it seems that they do have quite a lot of limitations, often because, the problem is just really hard. It's not really about the algorithms, it's about just the hardness of and the assumptions that you need to do in order to learn causal graphs from data. So then the question is just, what can you do? And, generally anyways, in biology, the end goal is not to learn this causal graph, right? It's like the types of applications that I told you about before. We want to be able to move cells from one state to another, or maybe, and oh, sorry, I do want to, this is an amazing place to be if you are interested in learning what has all happened in causal structure discovery. And they're just like all the experts in the room. So you should reach out to them if you want to, know what has happened in all these years. I'm just like giving basically, a very, very, very brief kind of overview of like where I think or why we have been thinking a little bit more about, what are the problems that you can maybe solve without actually learning the full underlying causal graph. Okay, but so what are these problems that we can maybe solve without learning the underlying causal graph, and what is the end goal? Okay, so so first of all, also, I mean, if you ask a biologist, or I have done this multiple times, what do they want? They will tell you they want this full 20,000, network on 20,000 nodes that explains the full underlying gene regulatory network. That's what they will answer, or have at least answered to me. But then if you ask, well, but what are you going to do with it, right? Then generally, or how are you even going to validate? Okay, I'll give you a network on 20,000 nodes, but then now how are you even going to tell me that you're happy with it? Well, then they would say like, well, I'm going to do this perturbation and then I'll see what changes and whether you can predict it using your causal graph, right? Well, then let me just go and do that problem, right? So if that's the problem that you want to solve, and generally, a lot of problems actually fall into this framework, is these two questions are really underlying basically, well, a lot of the biomedical questions we're thinking about so hard in the community is the following, is I have a whole lot of data where I did different perturbations. Say I did the genetic perturbations where I knocked out gene 1 and gene 2 and gene 3, but I cannot do all combinations, right? I cannot also knock out gene 10 and 20 and 30, and then next time gene, 20,000 and 3,000 and 3,001, etc. I'll never be able to do all combinations, even with a large scale data sets that we have. And I have also other perturbations like chemical perturbations. I threw this drug at it, I threw this drug at it, etc. So now from all this data that I give you in a particular cell type, say a particular cancer context, can you now predict what happens with all the other perturbations that I haven't performed? Okay, so can you, transport from some seen perturbations or interventions to other unseen interventions in the same context? So that's one question, right? That's underlying a whole lot of drug discovery, for example. Or, everything that I said about moving cells from one state to the other, because that's the reverse problem, right? I give you a particular state here that I want to reach. Well, can you tell me which perturbation I should do in order to reach that particular state? That's just a reverse question, right? Or there is the other transport problem, again, underlying a lot of what people are doing in pharma, is, I have all these different perturbations that were performed in the mouse. Well, what you really care about is what do these perturbations do in the human, okay? Or now with COVID, I mean, that's a huge transport problem, but another one is like, I have all these large scale drug screens that have been performed in one disease context, particular cancer context. Now here comes a new disease, which is COVID-19. Do I have to redo the whole drug screen, or can you now predict what actually this particular drug would have done in the new context, which is now COVID-19? Okay, so that's transporting across different contexts. Whereas here I'm trying to transport across different perturbations. So now the question is, of course, how much of this do I really need an underlying causal graph? How much of it can I maybe do with other types of models? And what is the right framework to think about these questions? And I'll add one more question here. So I put this in purpose because we have large scale perturbation screens that have imaging outputs like here. Every image is a cell. Okay, and you have, this is called cell painting. There are large scale, openly available perturbation screens where what you get to measure is, I do this drug and I give you a population of cells of how they look like. Or you can do this, which is gene expression. I do a particular perturbation and then what you get out is this 20,000 dimensional vector. But, you know, now I've been talking about gene regulation, right? And gene regulation always is this network on 20,000 nodes. And I want to, you know, and this kind of expression data gives me data on the nodes. And then from there, these are the causal variables. And from there, we're trying to learn the causal graph. But what about images, right? Here, it's not so clear. So, you know, this is, I mean, definitely something where we can do something causal because I give you a large scale perturbation data. But, you know, what are even the causal variables here, right? I mean, pixel 1 is certainly not a causal variable because pixel 1 in this image has nothing to do with pixel 1 in that image, right? But the causal variables are something that you have to learn. The causal variable could be, you know, and we have to learn it, but it could be how round is the cell, how round is the nucleus, how much of a protein is inside the cell nucleus, where is it localized? So where is this protein inside the nucleus, etc. Right? So, but we first have to learn what these causal features are, and then can we only do causal structure discovery if that is what we want to do. Okay, so there is a whole other question of like thinking about what are actually the causal variables in this whole system. So, so what we have is large scale interventional data, and what we have is very multimodal data. In particular, we have a lot of imaging data at the single cell level, and we have a lot of transcriptomics or sequencing based data at the single cell level. So how do we think about these questions? I first want to go into this question of like causal, what are even the causal variables? And then I'll go into actually these questions about transport between different contexts. Okay, so how do we even identify causal variables? And so maybe here representation learning can help. Okay, so this is work that we've done actually, and it was always motivated by trying to find something, or the intuition was that it should give us some causal information that we did here in biological contexts, and now have also some theory underlying it that I'll show you on the next slide. So same data as what I had before, just multimodal. So think of it, I have all these single cell images, and I have the same population. So I take a population of cells, say our T cells, our nice T cells. I take out some at random for imaging, and I take out some at random for getting the expression profile. Okay, so it's the same, the distributions are the same, right? Because at random, I chose out some for imaging and chose out some for sequencing. Well, now, how do I integrate both of these, and why should I even integrate both of these modalities? Well, let me first say what we did. So here, we're trying to learn one latent space, so one joint latent space for all of these multimodal data sets. So we have one autoencoder that goes from this imaging land to the latent space, and another autoencoder that goes from, say, sequencing land to this latent space. Now, the population of cells are the same, right? So I can add so first of all, so this is another challenge in biology. Generally, when you get a measurement, the cell is dead. Okay, so if I get an image from the cell, that means fixing the cell is dead. I cannot also go and get an expression profile. Or if I get the expression profile, I cannot then go and also get the image. So I'll never have paired measurements in the same cell. Okay, so, so here I do have measurements of many, many cells in imaging and many, many cells in sequencing, but I never have the paired measurement. But I know that the two distributions in the latent space should be the same, right? Because it's the same cells. So all we can do here is add, you know, say a classifier in the latent space that is, you know, penalizing me if I can tell whether the cell comes from imaging land or sequencing land. Okay, so that's how I'll make the two distributions in the latent space match. Now, for biology, you know, what is exciting here also is that we can then go from cheap imaging modalities to more expensive sequencing modalities. But I think what is more interesting and you know, what has motivated us is really looking at this joint latent space. And the intuition, and this is just intuition, that, you know, maybe this latent space should contain more causal information than what we could get just separately in this space or in this space. And why is that? Well, because, you know, whatever is causal should not depend on how you're looking at the system, right? It should be there whether you're looking at it from left or from right, or whether I'm looking at it from imaging land or from sequencing land. And so maybe, you know, invariance with respect to how you're looking at the data or how you're looking at the system could help you to get at whatever is actually the underlying causal features in this whole system. Okay, so that's just intuition. I'm not going to go, and you know, and there is of course now, there is now of course a lot of related work, but related work in the system of using invariance with respect to interventions, right? So invariance with respect to interventions, and invariance, and there it's invariance with respect to, in this causal graph, with respect to the causal effects. Whereas here it is invariance with respect to whether you're looking at it from modality 1 or modality 2, you're looking at whatever is preserved under the 2 modalities. And we've now also applied this to integrating, for example, heart MRI data and heart ECG data into a joint latent space, and then actually shown that you're much more powerful to identify genetic variants that are associated with a particular disease. Okay, again, suggesting that whatever this latent space is that we're getting is actually more causal than whatever you could get in each one of the different data modalities. So where do we go from there in terms of like actually the theory that we can for now say that this is, or where we got until now to say that something like this is actually true? And we had to wait for Kuhn to finally write out all the results that were always in his head, on which we could then build to get this particular theorem here, because Kuhn has done a lot of work in this field, and all this theorem here, if you read the paper, you'll see that we're very heavily building on all of his recent work that he all put out in 2022, one paper after the other on this. So that's really amazing. So what I'm showing you here is, and maybe some people even on the previous slide got this question of like, hey, maybe there are some variables that are only represented in the imaging space and some variables that are only represented in the sequencing space. Right here with our autoencoders, we actually made just one latent space that fully matched, that fully had to match both of these modalities. But here in the theory, and of course also practically, we now don't do that. We allow partially latent spaces that are not just fully overlapping, but can have also some separate latent variables. And here the theory is also like that. So we assume we have this, or what we would like to identify is this, latent. So every one of these Z's is latent, right? We don't know what the causal features are. We would like to learn this causal graph among these latent nodes. We have some latent nodes that are fully shared among all modalities. We have some latent nodes that can be just modality specific. So Z4 is, for example, only a causal feature for this modality, say imaging. These 2 latent nodes are only features for this modality, etc. And so from data on every one of the modalities, so say sequencing data, imaging data, etc., we would like to learn this underlying causal graph, right? This latent causal graph. And we would also love to learn what these causal features are, right? Is it that how much of this, like what is the shape, or how round or how ellipsoidal shaped is the nucleus, et cetera, Those are the features that we would love to learn in here. Okay, so that would be causal feature discovery. And so here I tried to put down, at least to also be a little bit concrete in terms of the results we have and a little bit more precise, all of the assumptions that we currently need to do in order to actually get this identifiability result. So for now, this identifiability result, what we need is that we have everything in here linear and non-Gaussian, so linear non-Gaussian in order to then allow application of linear ICA. And this is known as a mixing function, which goes from the latent graph down to the observations. So this function here is again linear. So literally everything is linear here. And what we need is the full column rank. So these are the shared latents, and these are the domain-specific latents. And then we have results that we actually have identifiability. So we can, first of all, and this is important for us, identify how many latent nodes we have, and we can identify the joint domain distribution. And then if you have additional assumptions on this, which edges you can have and not have, and this nice, assumption that came from Kuhn's work on how many children you need to have in every one of the domains, then you can in fact also identify what the underlying latent graph is. Okay, so very, very, we can only prove these kinds of things under very restrictive assumptions, but it suggests that, whatever we had here as intuition and why somehow these kinds of results, actually, these latent spaces are so important for us in biology, where we are really like always integrating all of the different data modalities into this latent space and then doing all of the downstream analysis like genetic association studies, etc., everything in this latent space. Okay. So now this is, of course, only one side of the story, right? Where every one of these, what we're measuring is just a different modality. So imaging, sequencing, etc. I haven't talked about interventions at all. right? So you can make the same story of like, I have one, and now, I won't have any separate, or for now at least, I don't have any separate latent nodes. Let me say I have a full latent graph, okay? So every node is latent, and I have some causal model among these latents. And now I'm doing interventions on these latent nodes. And what I get to see down here, so this X1, X2, X3 is now interventional distributions. Okay, so this would be an intervention when I'm doing, one perturbation, applying one drug. This is the data that I get when I apply a different drug. This is the data that I get when I apply a different drug, or I can also do genetic interventions. Okay, so now I'll change that. And so this is how we should think about it, right? So here we have our latent graph. We're going to do interventions on this latent graph, and I get my observations, these X1, X2, X3 for different interventions. And what we would like to do is actually learn this generative model, right? Of like, hey, I give you an intervention, and I give you observational data. Can you then generate how the data would look like if I was doing, adding this knockout and that knockout together? How does then the data look like, the interventional distribution look like? Of course, now we also don't, because these interventions are on latent nodes, we cannot assume that we know that this intervention affects that latent node, because that node is latent, right? So, so we need to figure out which interventions are attacking which latent node. So we need to find a grouping of the targeted variables in this latent, among all these latent nodes. And then, of course, we also want to identify this whole causal graph among these latent nodes. And again, what are the assumptions that we need? So here we actually need much weaker assumptions than what I showed you on the previous slide. So this function here doesn't have to be linear, it just has to be polynomial. The causal model is fully non-parametric. We have all kinds of faithfulness assumptions that we need. That's always kind of figuring out the right faithfulness assumptions is always the crux of all of this. And our interventions can be any soft intervention. And so then we do have this identifiability result that we have to have interventional data from at least one intervention per latent node. Then you can fully identify this causal model, identify which intervention targets which latent node, and then also the causal graph among it. Okay, so much weaker assumptions, and kind of the types of assumptions that we would want to see in results, not the types of assumptions that we unfortunately had to use here. Okay. Now, this is just a theoretical result. Now, can I also give you an algorithm that actually performs this? And so I just continued this theorem here, which says, okay, theoretically, we prove this, but also algorithmically, we do have an algorithm that, again, provably will actually give you identifiability. And so how do we do this now that I motivate? So we work a lot on autoencoders just because we understand the functions that they're learning. We have a lot of research on that. But here again is like a type of autoencoder that we're using. And so how does this work? So what I want to do, right? I want this generative process where I put in observational data, so unperturbed data, and I put in a particular perturbation, so say knockout gene 1. And what I would like it to spit out is how do samples look like when I'm knocking out gene 1, and give me samples from that interventional distribution, right? So so now I want to encode observational data, decode interventional data. And in between is this causal graph that we're trying to learn. This is this latent causal graph in here. But I also have to learn a representation of every intervention and figure out how does an intervention actually affect the causal graph, right? Which node is actually intervened on in this causal graph. And finally, this is the other thing that I also mentioned at the beginning, right? Whenever I get a measurement of a cell, the cell is dead. So I cannot get in the same cell the measurement of how it looks like in the observational state and how this particular cell looks like when you knock out gene 1. Right? So everything has to happen at the distributional level. So I do have observations without any knockout. And then I have observations with knockout 1. And now what I need to do here is, that's why we have this, this is a discrepancy-based VAE. We need to have this loss here, which is just a distributional loss, which says that our predicted interventional samples look similar, or our predicted interventional distribution looks similar to the interventional distribution that I actually see in the data. Okay, so all these results that many people have about always when you actually have paired data, meaning paired data in the no intervention and intervention setting, don't really work in biology. Okay, so we never have paired data. We'll never get to see like how the particular cell looks like before and after the intervention. So everything has to be done always at the distributional level. And again, here, there is a lot of really exciting research going on in this field of causal representation learning. In particular, we had this tutorial here by Danya and then by Jason already on Monday. And of course, a lot of work also on disentanglement, etc. So a really exciting field where I think there is still a lot of different open problems to work on. And, it's always nice if you can motivate it by a particular application where you can then actually show that it works in that particular setting, that you have the assumptions that, kind of match the particular application at mind. Okay, so that's, so now what I talked about is multimodality and different interventions, right? Now, let's go through this and see whether this actually solves any of the problems that I discussed. So let's see if any of this solves kind of the problems of like transporting from, different perturbations seen in the mouse to perturbations seen in humans, or transporting between different new interventions. Okay, so let's actually look at this particular model. So what this particular model can do is, for interventions that it has already seen, give me more data. Okay, so that's nice. It can also go in and say like, hey, I'm training this model on, knockout 1 separately, knockout 2, knockout 3, knockout 4, knockout 5. And then I can ask it, from among all of the interventions that I've seen, I can ask it for new combinations. I can ask it, right, if I put in here, a combination of knockout 1 and 2, can you predict how the distribution looks like under that combination of knockouts? Okay, so that's the kind of stuff. But what I cannot do in here is, I give you knockout data on separately knocking out gene 1, 2, up to 20. And then I ask you, hey, what happens when you knock out gene 1000? This approach cannot do that, right? Because I don't have a representation of what it means to knock out gene 1000. Okay, so generally what it cannot do is these types of questions where I'm giving you here data on knockouts of 1 up to 20, and then I'm asking you, hey, tell me what happens when I knock out a particular completely new gene. Okay, but that's what we'll need, right? Because I'll never be able to, or sometimes, in some contexts I can do knocking out gene 1 up to 20,000, but I'll not be able to do that in every cell type, etc. Okay, so now somehow there are questions that come up and about the difficulty of the different problems, okay? It's questions about the difficulty of like, how hard is this problem really? And I will not have any theoretical results on this, but like, is this a harder causal problem as compared to these causal problems? Is it harder to somehow, if I give you drugs, like these molecules, can you then predict the effect of new molecules? Is that harder than predicting, if I give you knockouts, can you then try to predict the effect of new knockouts? And first of all, I do want to point to, really nice work, and a whole, line of work by Elias Badenboim and Julia Pearl on causal transportability, where they actually solve these types of questions, but always assuming that you know the underlying causal graph. Okay, but of course in biology, we don't know the underlying causal graph. And I showed you all the problems of if we were to go and learn these causal graphs, how many mistakes we already do then, and then we could do as a second step this. So I would like to, if possible, shortcut that, right? That I don't have to go to learning the causal graph, then applying all these results, and then going to actually predicting the effect of an unseen perturbation. And so here is somehow the question that we have been asking ourselves the whole time, and where we're always moving back and forward. So, okay, so black box, so let's look at this particular question here, where I have particular perturbations and I want to predict what happens in a different context. So this, to us at least, and also of how we are applying it, seems to be somehow the easiest problem among all the three that I'm going to talk about, as compared to translating between different perturbations. Translating between different perturbations that are chemical structures seems to be easier than transporting between different perturbations that are genetic. And so why is that? Because we always have a prior somehow, right? So here I can see this as a matrix completion problem. Yes, it's not our standard matrix completion problems, but here I have a representation of drugs, right? And I have a representation of contexts. Because I have a lot of data, and if I want to go from mouse to human, I have a lot of data that represents me what is a mouse and what is a human. And I can represent drugs by protein, by the structure of the molecules. And, it's intuitive to, and we do know kind of that, similar molecules have a similar effect. Right, so I can somehow define similarities among contexts or cell types. I can define similarities among drugs. And based on these similarities, I should be able to solve some matrix completion problem. But how do I define similarities among knockout on gene 1 and gene 2 and gene 20? And then you give me gene 1000. Well, the similarity among these is actually given by the causal graph. Right? If gene 1000 is somewhere close in the causal graph to gene 1, well, then they will have a similar effect. But then I'm going around, so that means I do need somehow the causal graph, because that tells me the similarity among these different interventions at the particular nodes. So that to me seems like, at least intuitively and based on the different applications, always seems the hardest problem, where we really need something about the other causal graph. Maybe all the other problems can somehow be solved using, more standard representation learning kinds of problems. Okay, but I don't have any theory about it, but that's somehow where we're always going back and forth between where do we use just black box models and where do you actually use causal structural equation models. Okay, so here is how well black box do. So here I take this large-scale drug screening data sets that are available. You can take it in imaging land or sequencing land. This is called CMAP. So many, many thousands of different drugs, many different cancer cell types, or different, lung cancer, skin cancer, et cetera, et cetera. Every dot here means that this particular drug has been applied in this setting. All of this data is freely available, produced at the Broad. Anyone can look at it. And all the white space is where we don't have data, okay? But, since there is so much data, I can just leave out some of it and ask, hey, given all this data that you have, can you tell me how well you do on, predicting what this particular drug will do in this particular cancer? Okay. Now, these are causal questions, right? I'm trying to predict the effect of an unseen perturbation in a new context. But again, standard matrix completion approaches will not work, right? Because, I have here a full missing column. Well, standard matrix completion relies on low rank. So we'll just fill in a whole constant everywhere here. So that definitely doesn't work. But, more approaches where you think about similarity, about, and that actually leads me to synthetic controls and synthetic interventions, where you think about, hey, this particular drug is very similar to this other drug, and this other drug I have seen in this cancer context. Well, then maybe this drug will do something very similar in this cancer context. Okay. So that's so those are the types of approaches that have actually been quite successfully applied in these transporting between different contexts or transporting between different chemicals. So now what about genetic perturbations? And of course, genetic perturbations is kind of what we need in order to then ultimately solve the problem of moving cells between different states, because that's where we have, CRISPR can be scaled. We have all 20,000 genes that we can perturb in many combinations, etc. So how do we go about that, right? And for us, the intuition, like in order for me to say that this knockout will do something similar to that knockout, I do need something like an underlying causal graph that tells me they're somewhere close by in the space. And that's my representation of closeness. And so for these types of applications, we've always used somehow that we're trying to learn also some kind of structural equation model that represents how close two interventions are. And now I guess, there is a whole lot of research in here, and I will not go through it because I won't do it justice. But I think this is a really important problem that people should work much more on. Like, so and this is the problem. Okay, so I give you a defined state, which I want to get to. So I give you an interventional distribution. I give you the starting state. So that's the target. I give you the source distribution. Okay, these are my, skin cells. I give you the target distribution. Those are my neurons. And I'm asking you to find the interventions where the interventional distribution will actually give rise to this target distribution. Okay, so I'm asking you to find the interventions so that the interventional distribution looks like this target distribution. And so now what are the things that you can do? Well, so in biology, we can do this iteratively. Okay, I don't ask you to do that in one shot. Okay, would be great, but I don't believe you can. Okay, so, sure, you can do this iteratively. But all these like infinite horizon algorithms in machine learning, they won't do it, right? Because that works for online advertising, but actually performing these experiments does take quite a lot of time. So you might get 5 batches of experiments. Okay, so the batch setting is what we care about, not your infinite horizon setting. And you can perform a lot of interventions at the same time. So you can definitely perform, intervention on gene 1 and gene 10 and gene 20, et cetera, or combinations of them in every batch. That's totally fine. Okay, but I only give you around 5 batches of experiments. And, I do want you to not just learn something about the underlying causal system, but I actually would like you to move the cells to the state that I care about. Okay, so I do care about the target state. And, we've started to do things like where you have an acquisition function that is causally motivated, that, at least tries to find interventions that tell you something about the underlying causal model, and also try to move the cells towards the target state. And, we have, again, so here we have to make all kinds of assumptions, linear Gaussian structural equation model, that all of our soft interventions are just moving the mean. So those are these shift interventions, etc. Then we can come up with algorithms like this. But I think here there is still so many open problems that people should work on in the batch setting with other types of soft interventions, etc. And of course, again, a lot of related work in active learning, Bayesian optimization, bandits, etc. We've seen a great talk yesterday on active learning of particular soft interventions that was given here. But still, I think a lot of open problems. And with this, let me end. So I hope I was able to convince you that the biomedical sciences are not only uniquely suited to being one of the greatest beneficiaries of research in causality, or also more generally machine learning, but really one of the greatest sources of inspiration for it. And, some of the challenges and opportunities in biology is that we really have large-scale multimodal and interventional data sets, but we don't know the underlying causal model, and we also often don't know the underlying causal variables. Okay, so, what we need is some way of actually making the best possible use of all these observational and interventional data, in particular for problems of, how do I identify optimal perturbations in order to either move cells from one state to the other, and I can even do this in an iterative way. That's totally fine as well, but just in small batches of experiments. I've somehow always tried to combine representation learning and causality, but in some sense there is, or I've always asked myself whether this is in fact the right way of doing it. So somehow, when we're doing causal structural equation models, that's at this third level in the hierarchy, in Pearl's causal hierarchy. Whereas when we're doing representation learning on the standard methods like neural networks for representation learning, they're always down here. Right? And somehow to get at these questions of trying to predict the effect of an unseen perturbation, which is really level 2, we're just trying to somehow connect these two methods that are at the third level or at the first level. And maybe that's not the right way of doing it. Maybe we actually need a full new theory that is particular to the second level here, right? So, in some sense, we have concentrated a lot of identifying what are the differences, like how much harder are counterfactuals as compared to just estimating association. But there isn't much, at least to my understanding, sorry, too many understandings, of really understanding this difference here between the second level and the third level, or the first level and the second level. Okay, and what we really care about here is actually predicting the effect of an unseen intervention. So we really care about the second level because we are going to do the experiments. We don't care about the counterfactuals in this particular applications that I talked about. Okay, so the question is really, is this trying to marry representation learning and causal structural equation models that I've talked about to get at level 2? That's one approach, but might not be, the final approach. And maybe we just need a lot more theory that directly goes towards level 2 and tries to understand this. Okay, with this, let me end. I presented mainly work of 3 very talented PhD students in the lab. Jiaqi Zhang on all of the causal representation learning, Chandler Squires on structural causal discovery, and then Adit Radhakrishnan on everything that had to do with understanding autoencoders, etc. And then, of course, it's always very collaborative in the lab. So everyone has been part of some of all of these works. And then, of course, thanks to funding, without which it wouldn't be possible. And thank you all for your attention.
Thanks so much, Caroline. Some time for questions. We have a microphone, a couple microphones.
Can you give this is a great talk. Thank you very much. Can you give a sort of sense of why the soft interventions seem to be so hard to characterize if you have a different class of something than mean shift? What's the technical difficulty that's there that's preventing you from predicting what would happen under such interventions?
Yeah, so let me go to this. So these would be, and I'll actually take your question a little bit different because what I meant is maybe a little bit different. So mean shift is easy to deal with, and that's why we have so much work around it. But I think it's not the relevant one. And we've done so much of it until we understood that it's actually not relevant at all for these biological applications. So when you add these transcription factors, we thought we're just shifting the mean. But actually, if you look at the data, it looks like a hard intervention. So meaning that it's not just a mean that shifts, but actually the variability is going down a lot. Okay, and now it's clear why that's the case, because when you add these transcription factors, you're adding them basically at infinity. So you're adding them to such a high level that the cell has never seen. So it's the same as kind of doing something like that we would think of as a hard intervention that really then reduces the variability. And I don't think we have the right theory to think about, like first of all, come up with a right model of soft intervention that is relevant here. And then think about how that affects the algorithms.
Okay, but there's no like technical problem that makes it hard to model any specific kind of soft intervention that you've encountered?
Yeah, I just don't know which one is the right one. Okay. Yeah, I don't even have the right model yet. And we also like shift interventions, actually even, I mean, we now like discovered this in some, like it even gives you more identifiability than your standard interventional Markov equivalence classes. So I also don't think we have like that fully characterized. We always think of interventional Markov equivalence class, but actually it depends a lot on which intervention you're doing. Thank you.
Daniel, can I ask a question? I'm online.
Sure. Yes.
Oh, great. Yeah, this is a really great talk. I really appreciate, many things we do is similar to what you're interested in as well. I have two comments or maybe question and comments on the, one is the perturbation representation. I think that there's a certain way you can learn the representation. For example, maybe more is on the cancer. So maybe I'll reverse the question. So first, as a perturbation, I noticed that you mostly try to use in a systematic perturbation, but not wonder whether you pay attention to the TCGA data, which is a perturbation of the nature by nature and causing the cancer. And so that perturbation data is more interesting than the systematic perturbation, because you always have a chance you perturb something, a gene that is not utilized by a particular cell, so you do not have impact. Or maybe if you randomly perturb the cells, the cells could go anywhere. So you do not have a sufficient pattern to really find the impact of different perturbations, whether these different perturbations share a common impact. So that's what. So, but if you study,
let me just shorten it up. So you're talking about genetic perturbations that are available and say that are actually happening in cancer cells. I'm actually talking about cell engineering. So I do want to identify. I do want to move cells outside of the state that they're currently in. But that doesn't mean that, thinking about what are the perturbations that actually just happen in cancer directly is not an interesting problem. It's just not the problem I talked about here.
I understand. Yeah. Okay. Yeah. So in that sense, we try to learn.
Sorry, we're running out of time. I'm being told.
I'll catch up with you.
In the room, there's one in the back there. Yeah.
Hi. Thank you for an amazing talk. So my question is related to the inherent trade-off that you were talking about while you're trying to get to the best intervention. At the same time, you don't know the graphs, so you want to learn it as well.
Yep.
So specifically in the case of genetics, what would be the right assumption? For example, what kind of operations are costly here? Is it that the number of nodes that I can intervene on bounded? Are observational samples costlier than interventional samples, or they're almost equal? So yeah, that's my question.
Yeah, those are really great questions. So what is costly is getting an observation, independent of whether it's interventional or observational. Just sequencing a cell has a cost. Generally, all these interventions, we actually already have libraries that are set up. So you do this via viruses, and we already have a virus that knocks out this gene, a virus that knocks out that gene. So all of that, you can remove the costs. So it's literally about getting an observation, whether it's interventional or observation. So per cell.
Okay.
$1 per cell is basically how you should think.
Okay, so I think we're probably.
Hello. Yeah, thank you. That was a nice talk. So I have a question is like, when you have the causal graph of the latent variables, like how do you get meaningful information about from that? Like what does that mean to intervene on a latent variable?
Yeah, those are really great questions. So what we're getting out of this is, first of all, that if you're thinking about a drug, that we're basically, for example, what we get out of it is clustering drugs by the drugs that have the same targets, right? So yes, they're latent, but because now I can tell that all of these drugs are targeting the same latent variable, then I know that all these drugs have the same target, which I generally wouldn't know. So that's, for example, something. So these grouping of targeted variables is something very interesting that you would get out of. And then, of course, here, what we would really like to do is like actually get the meaning of what these latent variables are. Like what I said, like, is it a lot of protein inside? Is it the shape of the cell, etc.? Does that answer your question?
Great talk. Thank you very much. Is there anyone else?
Hello?
Yep.
Okay. So I have a question about the multimodal representation learning. Specifically, if I understood this correctly, then you incentivize the neural network to learn the same latent distribution for the different modalities. And I'm a bit confused why this is interesting. Wouldn't you really want to have a mapping between like the same data points corresponding to the different modalities? I understand that you don't have paired data points, but I'm just trying to understand why this is interesting to just have the same distribution.
Yeah, I think that's a great question. So, and I agree with you that that's why here we already extended it, right? That not the whole latent space is shared, but you do have domain-specific latent variables. Because I do think that, you know, in every modality, there are some latent variables that are not captured by the other one. But for sure, there will be something that is shared because it is looking at the same cells and the underlying system in those cells. And, you know, generally here, I mean, this was just a first approach, right? We mapped to the same latent distribution because we also wanted to translate. So, you know, some of these modalities, and that gets to the cost. So imaging is much cheaper than sequencing. So this was pretty a surprising result for biologists that, you know, you could take these very, very cheap modalities and I can in fact translate to these very expensive modalities. And now we have more and more proofs about that we can really do this. So even the fully shared latent space actually captures enough information for us to be able to translate for biologically meaningful, say, clusters, etc.
Okay, interesting. Yeah, thanks.
Thanks again. So in the last part of the work, you try to go from prediction to control, right? So in the last part,
yes.
Essentially, you have two distributions, right? Where variables are given. You want to have, in some way, you like to have a minimum number of changes in the direct causal influences to move the distribution, right?
Yep.
So from this perspective, do you think we can directly do something like a causal discovery from non-stationary data or heterogeneous data in order to really identify where to apply the minimum changes?
Yeah, so I'm hoping that something like this is exactly what you should be doing. And minimum change is maybe not, I mean, yes, you do want small change, but I don't care about minimal. So even if you have
number of target variables you want to change directly.
Yeah, but I don't care so much, right? So I don't really need, like, if you can do it with 3 or 4 or 10, I think I'm very happy with that. So you don't want to do it too many because generally the cell dies if you do too many perturbations at the same time. But I wouldn't minimize. I care more about getting like as close as possible to the target distribution.
Cool. So I think we could probably have Caroline here all day answering questions, but it's time for a coffee break. So let's give her another round of applause.